Thank you so much, Peter, for the kind invitation and for the very nice introduction. So, let's see what the current state of play is for the seizure detection systems. These are my disclosures. And this is the outline of my presentation. So, first I will discuss with you why we need seizure detection using wearable devices. Then we'll discuss how we know that they actually work. I will show you validated devices, and I will tell you about the clinical practice guideline which has recently been issued in collaboration between the International League Against Epilepsy and the International Federation of Clinical Neurophysiology. So, why do we need automated seizure detection? Well, we need that for intervention and prevention, because we know that seizures may lead to injuries, and generalized tonic-clonic seizures, the big convulsive seizures, are the major risk factor for sudden unexpected death in epilepsy, SUDEP. We want to prevent that. Then we could use automated seizure detection in closed-loop systems to trigger on-demand therapy. And then very importantly, the unpredictable nature of seizure occurrence is distressing and disabling both for the people with epilepsy but also for the family members, for their caregivers. And this may lead to social isolation and of course decreased quality of life. And we know that roughly half of the seizures remain unreported by the people with epilepsy. And what's the consequence? Well, of course, that the therapeutic decisions, but also the drug trials, are largely based on unreliable data. And of course, this applies mainly for the nocturnal seizures, where 85% remained unreported by the people with epilepsy, but also almost 1/3 of the daytime seizures remain unreported. And of course, this is mainly the so-called focal impaired awareness seizures, but also the big convulsive seizures, almost half of them remain unreported. So we need such devices, and there are several published large-scale survey studies interviewing people with epilepsy, their caregivers, but also the healthcare professionals, and everyone says, yes, we need reliable seizure detection devices. So that's why our community is working intensively on developing and validating such devices. And if you just search PubMed, you can see a huge number of publications, really many papers about seizure detection, but do they really work? And what is the level of evidence that these devices actually work? Well, if it comes to a drug therapy, then you can rely on the big multi-center prospective randomized controlled trials. You know that they are conducted in a proper way, and then you can rely on those results. But when it comes to diagnostics or even seizure detection devices, it's quite a mess, or it used to be quite a mess, because everyone was publishing it. This works, that works, without real criteria, without clear guidelines how to test them. And we tried to overcome this, and my colleague and friend Philippe Rivlin and I, we suggested how to validate the devices, and then we took input from the so-called QUADAS-2 criteria, which is a systematic way of evaluating the evidence in diagnostic trials. And then we tried to apply that for the seizure detection devices and construct 5 phases from 0 to 4, exactly as for drug trials. So phase 3 for a drug trial and also for a seizure detection trial, in our interpretation, provides you with compelling evidence that this device can be trusted. This device works indeed. And then phase 4 is an in-field study where we test whether this works also in the home environment of the people with epilepsy, because most of the trials are done in epilepsy monitoring units, which are kind of an artificial environment. Now, we used this criteria when we evaluated the published evidence for the clinical practice guideline. So this has been developed by a working group approved by both societies, the Epilepsy Society and the Neurophysiology Society. And we used a very rigorous methodology for guideline development. So we had a systematic literature review, and then we used the grade method to weigh the evidence. And then our proposal, guideline proposal, was out there for public hearing, for public comments for 2 months on the homepage of the International League Against Epilepsy. And very importantly, it was also reviewed by the IBE, the patients' organization. And then in the end, it was endorsed by both societies. So here you can see that we started by reviewing more than 1,200 publications, and then we boiled them down because we only wanted to evaluate those which have a higher level of evidence, like phase 2, 3, and 4 trials. And this boils it down to only 28 studies. So out of the 1,200, only 28 complied with these more rigorous criteria. And this is the conclusion. So when it comes to seizure detection, we only have compelling evidence for the big convulsive seizures, for the generalized tonic-clonic seizures, including what we call now focal to bilateral tonic-clonic seizures, so seizures starting focally and then leading to a generalized convulsion. So when we did the literature review, there were 3 studies which qualified for phase 3. And since we published the guideline, 2 more were published, but this would not change, in my opinion, the conclusion in the guideline. Now, phase 4 studies, when we published the guideline, we found 2, and 2 more were published since then, but they point into the same direction. Now, they all say that for these big generalized tonic-clonic seizures, the devices that are out there can catch them with a sensitivity between 90 and 98%, depending on the device, and then with a false alarm rate between 0.2 and 0.7 per day, but fortunately relatively low during the night, because that's the most disturbing period when a false alarm can occur. Fortunately, during the night, this is rather low. Now, for the other seizure types, we can call them non-convulsive. For all the other seizure types, we only found a lower level of evidence, only phase 2 studies. And since the guideline was published, a single phase 3 study was published for absent seizures. I will come back to that later. Well, the sensitivity was not bad, so some of these devices achieved a sensitivity of 90%, which is not bad, but this came at a cost of a very high false alarm rate, up to 500 false alarms per day, which I think you would agree with me, it's totally unacceptable. Nevertheless, there are promising results out there for variable EEG, variable EKG, heart rate variability, so there's a lot of research going on. But for the other seizure types, other than the big convulsive seizures, we are not there yet. Now let me show you a couple of devices which have been validated in robust phase 3 trials. And probably the mother of all seizure detection devices, wearable seizure detection devices, are those based on accelerometry. So these are movement sensors. Here you can see this device. Let me see, where's my cursor? There's my cursor. Can you see my cursor? Yes. So this is a kind of a wristwatch with an accelerometry device there. And here you can see in 3 colors, in the 3 dimensions of space, how a generalized tonic-clonic seizure is perceived by this sensor. And then there is a Bluetooth connection between the device and a smartphone. And the nice thing about this device is that it works also outside the home. So the people with epilepsy can leave their home, and an alarm is triggered and is sent using the smartphone, and also with a precise localization of where in space the person with epilepsy is situated when this occurs. Well, the sensitivity is 90%. The mean detection latency is a little bit long. It's 55 seconds after the seizure starts. Why? Because this is triggered mainly by the clonic jerks, and those occur in a later phase of a tonic-clonic seizure. Nevertheless, the false alarm rate was low, only 0.2 per day. Relatively low, of course. This is another seizure detection device which we have developed in collaboration with Denmark's Technological Institute. This is based on the signals from the muscle. So there is a mini EMG, mini muscle detector here. It's a self-adhesive patch. with a mini EMG machine which records the signals from the muscle, and then we designed an algorithm which detects the generalized tonic-clonic seizures with a sensitivity of 94%. And here the median detection latency is shorter. It's only 9 seconds because this is triggered already in the initial phase, in the tonic phase of the seizure. The false alarm rate was higher than we hoped for, 0.7 per day, but fortunately very low during the night, 0.01 per night. So this device can do something else than just triggering an alarm with a seizure. It also can differentiate between an epileptic generalized tonic-clonic seizure and a psychogenic non-epileptic seizure with quite a good accuracy of 95%. This is another device which is multimodal. That means that it's triggering the alarm based on different modalities. And you can see here it's called Night Watch. It is based both on accelerometry signal, as the first device I showed you, but in addition, it also combines data from measuring the heart rate. Now, its use is restricted to nocturnal. That's also the name, Night Watch. And for the big convulsive seizures, generalized tonic-clonic seizures, it detects them with a sensitivity of 81%, but it also detects other nocturnal major motor seizures, but with a somewhat lower sensitivity, only 77%. The false alarm rate, that's relatively low, only 0.03 per night. This is another device which is multimodal. It combines, again, accelerometry with electrodermal activity, which is a measure of the autonomic nervous system changes, which are quite often during seizures. It also targets the big generalized tonic-clonic seizures. So this has been tested in probably the largest trial for wearables, and the sensitivity was quite good, 98%, with a false alarm rate of 0.84 per day. And very importantly, during the rest period, 0 false alarms. That's good. Unfortunately, the median detection latency was a little bit too long, 37 seconds, but this is because much of the input of the algorithm comes from the movement sensors, which are activated mainly in the later phase of the seizure. This is indeed a very good performance. Now, since the guideline was published, we also developed an automated computer vision. So this is based on an artificial intelligence algorithm, which in real time can analyze the movements. And we trained the algorithm to detect seizures. Unfortunately, it works only for the generalized tonic-clonic seizures and for the big other big convulsive motor seizures like hypermotor seizures, but doesn't work for the other seizure types. And even for these big convulsions, the false alarm rate is quite high, is 0.16 per hour, which means that this, as it stands currently, it's not really useful in a home setting, but for example, in a protected home where you have nurses surveying patients, that could be a help for the nurses, but the number of false alarms is too high for use at home. But we are working on it, and we are trying to improve it. Now, these were phase 3 studies. These were all conducted in the epilepsy monitoring unit. Why? Because we needed a ground truth, a gold standard. And where do you get the gold standard? Well, from doing video EEG recordings. But again, that's an artificial environment. Can we extrapolate these data to the home environment of the people with epilepsy? Well, this is the question which are addressed in the so-called phase 4 studies, the in-field studies. And here we asked the people with epilepsy, these were mainly in protected home facilities, how useful it is, and on a 7 scale, the median was 5.5. The performance was very similar to what was in the home environment of the patients to what we seen in the epilepsy monitoring unit. And then the caregivers and the people with epilepsy said that more than half of them said that the alarm helped them to log the seizures into their diary, and then 40% of them said that the alarm helped them to prevent injuries. And this is another in-field study with a baseline period and an intervention period with the night watch. It showed nicely that the level of stress of the caregivers decreased significantly. Unfortunately, they did not reach a significant level for the quality of life. This was another in-field study where we conducted online interviews with patients using seizure detection devices. And again, it was a high retention rate, so people kept using it. Overall, they were satisfied with it. It increased the number of seizures they logged into their diary. In 30% of cases, it helped decreasing the injuries. And also the validated devices perform better, significantly better than the non-validated devices. So would this help to prevent SUDEP? Well, we only have indirect evidence for this because we know that 91% of the patients die at home, living alone. They are found in bed in a prone position. And the people with epilepsy without nocturnal surveillance, not sharing a bedroom, and experiencing tonic-clonic seizures have a 67-fold increase in the risk of SUDEP. Now, there is a very important aspect. So the alarm is not sufficient. You need someone to intervene when the alarm is triggered. And this is shown by the sad story of a 20-year-old patient who wore a wrist sensor seizure detection device. A generalized tonic-clonic seizure occurred at 8:50. A caregiver trained in giving cardiopulmonary resuscitation arrived 15 minutes later, found the patient pulseless, prone, facing pillow with mucus in his mouth. He tried to resuscitate him without success. So 15 minutes is too long. The intervention must come quicker. Now, how about the other seizures? Well, of course, we can catch them with a full array EEG, but you agree with me that people with epilepsy would not like to go out on the street wearing full EEG hat. And when we ask people with epilepsy, they don't really want to get an implantation just for the sake of seizure detection. So what can we use? We can use EKG because the heart gives a huge electric signal. And then we have promising data, only phase 2, but promising data that we can catch seizures using the heart rate variability. This is not good for all people with epilepsy, but only for those who have a marked autonomic change during the seizure, but this is roughly half of the people with epilepsy. And now we recently have implemented our algorithm also in a loop recorder, which is routinely used by the cardiologist. This is a small subcutaneous EKG, which can sit there for months, so we have promising results with this. So whereas you can hide electrodes well in the ear, but unfortunately that would catch you only generalized tonic-clonic seizures. Another study showed promising results with behind-the-ear electrodes, but we are not there yet. And then subcutaneous EEG, again, promising results, but we are not there yet. Now, absent seizures can be quite well detected with this headband, but again, the false alarm rate needs to be improved. So we use the GRADE method to weigh the evidence, and this is what the two societies recommend. Using clinically validated wearable devices for automatic detection of generalized tonic-clonic seizures when significant safety concerns exist, especially in unsupervised patients who do not share a bedroom, but where alarms can result in rapid intervention within 5 minutes. And this is a conditional weak recommendation, but we cannot issue any recommendation for the other seizure types. And there is, of course, need for further research and development to increase the performance of these devices and to decrease the false alarm rate and to conduct properly designed clinical validation studies. And we need in-field studies. So we must prove that this makes a difference for the people with epilepsy. So to sum this up, we can reliably detect generalized tonic-clonic seizures using these validated devices based on movement sensors, muscle signals, and the multimodal and the video analysis. And there are promising results for the other seizure types, but we are not there yet, and everything else is still in development. Thank you for your attention.
Thank you very much, Sándor. We do have a couple of questions here, just to put it to you. So, do seizure detection devices still work for people with movement disorders, for example, essential tremor or Parkinson's?
Well, I know that there are several studies using wearable devices for movement disorders. They also use it for quantifying the improvement during trials. So a higher and higher number of drug trials are using these measures from wearable devices as an objective measure of improvement. But I'm not an expert in the movement devices. I'm working mainly with epilepsy. But yes, they are working on it too.
And the other one, I guess it's asking what type of device is going to be the leading ones in your opinion. I'm not sure if that means accelerometer-based ones, or I guess maybe what's the future look like? Is there a role? We're hearing AI everywhere as well. Is there?
Okay, so my response is that one size does not fit all. I mean, as we give specific drug to the individual person with epilepsy, the same way we will tailor to the individual patient the best device. So someone who has many generalized tonic-clonic seizures, obviously an accelerometer is good, but for someone with just a focal impaired awareness seizure, then we would go for either an EEG-based or EKG-based device. So I think in the future we will tailor the seizure detection device to the individual person.